I will start in 2-3 minutes. I'll just wait 2-3 more minutes and then start. Right, I'll share my slides. So this is the 11th live session of this course, Structural Biology. As always, I'll first be discussing the topics covered in week 11, and then we'll go through some practice problems. So we know that proteins are large macromolecules that are comprised of one or more long chains of amino acids. Proteins differ from one another primarily in their amino acid sequence, which is dictated by the nucleotide sequence in their genes, and which usually results in protein folding into a specific 3D structure that determines its activity. So we know that from DNA, a process called transcription converts it to RNA, and a process called translation converts it to the protein. So depending and three RNA, three nucleotide, three nucleotides code for one amino acid. So as I was saying that from DNA, a process called transcription converts it to mRNA, and from there, a process called translation converts it to proteins. Now, depending on the nucleotide sequence on the mRNA, the protein sequence of amino acids is determined. And from different sequences, we get different 3D structures. And depending on the structure, the activity of the protein is determined. Now, improvement in the activity and usefulness of an existing protein or creation of a new protein function by making suitable changes in its amino acid sequence is called protein engineering. That is what we will be covering in this class. Protein engineering utilizes rDNA technology, that is recombinant DNA technology, to introduce the desired changes in amino acid sequence of enzymes. So how does that work? We saw it briefly before also. So we have a DNA containing the gene of interest, and we have the expression system that is plasmid. we will remove a certain region of the plasmid and insert the DNA containing the gene of interest. Then this modified plasmid, known as the recombinant plasmid, is inserted into the expression system, which can be E. coli, and then it is expressed so that the new gene is now being read and expressed in the expression system. So this is the recombinant DNA technology, by which we, instead of the native gene, whatever is present, we can insert the gene that we want, so that a mutated protein is produced from the new gene instead of the native protein. In addition, increase in the level of production of protein is also part of protein engineering. So it can be that like this, using this method, a lot of the protein is expressed very quickly. This is a young discipline with much research taking place in understanding of protein folding and recognition for the protein design principles. So this is a very up and coming field which is very useful in biology today, that is protein engineering. So we know that amino acids are the building blocks of proteins, and each amino acid has an amino group, a carboxyl group, and a hydrogen. There is also a R side chain, which is of 20 different types for the 20 naturally occurring amino acids. Two amino acids can come together and a molecule of water is lost. So from one amino acid, H is lost from the amino group, and from another, OH is lost from the carboxyl group, to form a peptide bond, that is C double bond O NH bond. And like this, amino acids attach one after the other to form long peptides connected by the peptide bond. The second level of organization is the secondary structure, where the chain of amino acids is arranged in stable structures, which can be alpha helix or beta sheet, which are stabilized by hydrogen bonds between the carbonyl C double bond O and amide NH. The third level of organization is the tertiary structure, where different two secondary structures come together and are folded together and held by side chain interactions. And from there we get the quaternary structure, where more than one amino acid chains are held together by hydrophobic interactions or other non-covalent interactions. Sometimes instead of tertiary structure, you can have super secondary structure, which is basically another kind of tertiary structure, where you have different secondary structures arranged together. Like you can have four helices arranged together in what is called a four helical bundle. Or you can have alternate beta strand alpha helix, like this. The blue is the beta strand, red is the alpha helix. Alternate beta alpha structure, which is called Rossmann fold. So super secondary structure, you can consider it as another type of tertiary structure. These are the different levels of protein organization. And we have studied this before also, that when you have a chain of amino acids, there are certain dihedral angles which become important. So you have the dihedral angle which starts from C carbonyl of one residue. So you take the carbonyl carbon, nitrogen, C alpha to the carbonyl carbon of the next residue. So basically you start from here, come to N, go to C alpha, and again come to the carbonyl carbon of the next residue. This rotation is called phi. And if you start from nitrogen, let me just start from this nitrogen, again to C alpha, then to the carbonyl carbon to the next nitrogen. This rotation is called the psi. So these are dihedral angles that are important when and have specific values in different secondary structures. Another dihedral angle is from one C alpha to the next C alpha. So you have one C alpha, carbonyl carbon, nitrogen, next C alpha. This is the omega dihedral angle, and you can see this is 180 degree when it is S trans, and it is 0 degree when it is S cis. S cis is generally the more unstable conformation, because there is steric repulsion between the substituents on C alpha, whereas S trans is the more preferred sterically favored conformation. You can also have different dihedral angles in the side chains. So this is a long lysine side chain. You can see lysine C alpha, beta, gamma, delta, epsilon, NH2. So rotation along the different side chain bonds can be labeled as chi1, chi2 and so on. We had also gone through Ramachandran map, where basically the phi and psi dihedral angles I discussed here were plotted, and different regions of the Ramachandran map correspond to different allowed secondary structures. So all the white regions are disallowed regions of the Ramachandran map, meaning at these phi psi angles there is too much steric repulsion, and so these phi psi angles are not allowed. The red regions are the well allowed stabilized place. This is the alpha helix helical region, this is the beta sheet region, this is the left-handed alpha helix, and the yellow regions are the border space, meaning that they are allowed, but they are not as well allowed as the red regions. So you have different turns also occupy different regions and so on. So the Ramachandran map gives us an idea about which kind of secondary structures are allowed and which are disallowed. Now let us come to how a protein can mutate. So all the factors which are responsible for adaptation, optimization and innovation in the living world are proteins, majorly enzymes. Evolution executes a simple algorithm of diversification and natural selection, an algorithm that works at all levels of complexity, from single protein molecules to whole ecosystems. Let us look at what this means. So suppose you have a native protein having a stability of minus 10 kilocalorie per mole. This is the native protein. Now, as the organisms are evolving, there is some mutation in the protein which is destabilizing it by 3 kilocalorie per mole. So instead of minus 10, the mutated proteins are at minus 7 kilocalorie per mole. But there is a threshold of stability in which the protein can still function, it can still exist. And let us assume the threshold is minus 5 kilocalorie per mole. So the mutated protein is still within the threshold allowed stabilization. And so the mutated protein will continue to reproduce and evolve in the next generations. So in the next generation, you can have a series of organisms with having this mutated protein of minus 7 kilocalorie per mole. Now, suppose there is another mutation which is destabilizing it by another 3 kilocalorie per mole. So now it is crossing this threshold and coming to minus 4 kilocalorie per mole. This is not within the allowed threshold, and organisms having this protein will start to die out, because if the protein is not stable, it will not be able to fully carry out the functions required of it. And so organisms having that protein will die out faster by natural selection. So this is what is happening. Diversification is happening where the initial native protein is mutating in different ways, and it is followed by natural selection. So if a protein crosses the allowed threshold of stability, by natural selection, that organisms having that protein is dying. And so that unstable protein is not carried on to the next generations. So this kind of evolution happens in nature. Now, the mutation in protein can be of different types. One is a mutation which results in misfolding. So suppose there is a native state of the protein, but due to the mutation, now the three-dimensional structure is different. And this misfolded protein can in many cases have aggregation. So what will happen if aggregation is there? The protein will not be able to function properly. It will just aggregate with other protein molecules and precipitate out. So in this case, the organism will die out. Or there can be a change in the surface of the protein. So suppose the initial protein was able to bind with this ligand, but due to the mutation, the surface of the protein has changed, and now it is no longer able to bind to the native ligand. But sometimes what can happen is serendipitously, the new protein is able to bind to a new ligand. So this is called misinteraction, where a new interaction suddenly happens with the mutated protein. In these cases, often the mutated protein can have a completely different function compared to the original protein. Third is a mutation which is neutral mutation, that is, it has no effect. The original protein was also binding to this ligand, the mutated protein is also binding to this ligand. In that case, the mutation has no effect, just the protein is slightly changed structurally, but it is a neutral mutation because it is having the same function as the original protein that was binding to this ligand. These are the different kinds of mutations. Now we will again briefly reminisce what we have learned earlier about polymerase chain reaction and Sanger sequencing. So in any of these studies, along with a lot of protein, we need to have a lot of the DNA of interest as well. So how from a small amount of DNA we can very quickly replicate many copies of the DNA is the polymerase chain reaction. So the first part is denaturation, where the two strands of DNA are separated out into single strands by heating it to 95 degrees Celsius. Now, each of these single strands will behave as a template on which the new DNA is formed. The second step is called annealing, where from 95 degrees, the temperature is reduced to 55 degrees so that the primers can start annealing to the template DNA. This is required because the DNA polymerase can only add a nucleotide when there is a pre-existing 3' OH group. That is why it was essential first to denature the native DNA. The third step is called extension, where the temperature is again raised from 55 to 72 degrees Celsius and the DNA polymerase begins adding nucleotide at the end of the annealed primers. So after annealing, you have the initial primer added to both strands. You can see shown in purple here. And this is followed by the third step called extension, where at 72 degrees Celsius, one by one complementary nucleotides start adding and the DNA gets extended. So at the end of one cycle, from one DNA, you will have two DNA. At the end of second cycle, you will have four DNA. So on, at the end of n cycles, you will have 2 to the power n copies of DNA. So this is a technique by which very rapidly so many copies of DNA can be formed from just the original DNA. The other is called Sanger sequencing. Here what happens? It is polymerase chain reaction, but there is a modification. Instead, normally in polymerase chain reaction, we have a mixture of nucleotide phosphates, which add on to the template strand. Now, along with the DNTP, DDNTPs are also there, which are dideoxyribonucleotides. So if you look at the normal ribonucleotide, that is DNTP, and the dideoxyribonucleotide, the difference is that in the normal DNTP, there is a 3' OH, which helps to extend the strand. So when one nucleotide is added, the one after that can add on to it because of this OH group. But in dideoxyribonucleotide, this OH is absent. So once the dideoxyribonucleotide adds onto the chain will terminate there and there will be no further extension. So if we carry out polymerase chain reaction with a mixture of DNTP and DDNTP, we will get a variety of DNA strands, some of which may be the original replicated DNA, but others will be the DNA terminated at different lengths. Depending on where the DDNTP is introduced, we will get different DNA strands of different lengths. So whenever DNA polymerase is incorporating a DDNTP at random, the chain extension ceases. And so we will have many copies of the DNA of interest terminated at random lengths. After that, the random DNA lengths are separated by capillary gel electrophoresis, where larger fragments move slowly, whereas smaller fragments will move fast through the gel. And finally, by laser excitation, it is detected on a sequencing machine. So that will help you understand the sequence of the DNA. So Sanger sequence is basically used to find out the DNA sequence when you have a unknown DNA. You can just first denature the DNA to get the two strands, Then in the annealing process, add both the ribonucleotides and the dideoxyribonucleotides, where you have no OH at 3'. That will give you many copies of the DNA, some of which are the full DNA and some of which are fragments of the DNA terminated wherever the DDNTP is inserting. Then the fragments can be separated by capillary gel electrophoresis, and they can be put in a sequencing machine to find out what is the sequence of the DNA. Now, when we design proteins, what are the things we want to focus on? One is so when we are designing a protein, what are the things we want to focus on? One is the folding of the protein, that is the structure, determined by thermodynamic stability. So you have a protein in the native state, and on the way to the unfolded state, it can have many different local minima. So the native state of the protein is the global minima, but on the way while it is folding, it can occupy many local minima, which can be different transition states. So there is an equilibrium that is present between the unfolded and folded state of the protein, and we can perturb this equilibrium by mutating the protein. We can also want to focus on the thermal and environmental stability, or the temperature, pH, and so on. So by different protein mutations, we might want to focus on what is the stability of the protein and the equilibrium between the unfolded and folded states. Another thing that can be focused on is the function of the protein. So suppose we will learn all these techniques in the upcoming slides, but suppose we design a mutated protein by different methods, which we will discuss subsequently, and then we screen the mutated protein for its function. Sometimes the mutated protein will bind with the desired ligand with much higher binding affinity than the native protein. So that will be a positive mutation. So by mutating the protein, we can increase its binding affinity to the target ligand. Sometimes we can even change the binding affinity and make it bind to a completely new ligand. So there are different ways. These are the different things a person might want to do while they are doing protein mutations. Another thing is the catalysis. So a new protein can be designed which has increased catalytic rates. So if the knowledge of the catalytic mechanism is present, that is, we know that where the hotspot is, where the enzyme is binding with the reagents. Then by mutating the hotspot residues, we can increase the catalytic rate and control the rate of catalysis. So that is another aim that a person might have while carrying out this protein engineering. Now let us look at the factors which affect the stability of a protein. One is the hydrophobicity. So hydrophobic interactions, when you have so aliphatic side chains like leucine, valine, isoleucine, they can have hydrophobic interactions, which are basically non-covalent van der Waals interactions. There can be electrostatic interactions or salt bridges. So if you have a NH3 plus and COO minus, there is a stabilizing electrostatic interaction between them. Like in a helix, if you have a glutamic acid and a lysine at I plus 4 positions, there will be a stabilizing electrostatic interaction between them, because glutamic acid will have COO minus and lysine will have NH3 plus. So the opposite charges will stabilize the helix. You can also have hydrogen bonds, like here between C double bond O and OH. There can be disulfide bridges. So like here, you can see two sulfurs are connected, or there can be metal binding. So there can be a metal ion which is stabilizing this fold. So metal ion is positively charged. Suppose there are two acidic residues here, like aspartic acid or glutamic acid. They can, this is also an electrostatic kind of interaction of the negatively charged side chains with the metal ion. Another can be reducing the unfolded state entropy by mutating some amino acid residue to proline. So we know that whenever a protein is folding from unfolded to folded state, entropy is lost. So that is, thermodynamically, entropy is lost when a protein is folding. But if proline is present, proline already has a restricted entropy because of its cyclic nature. And proline has very restricted conformations, as we saw from the proline Ramachandran map. So if we mutate an amino acid to proline, the loss in entropy when it goes from unfolded to folded state is also reduced. So that way also the thermodynamic favor towards folding can be increased. So these are different interactions that stabilize the folding of a protein. Now we will look at some protein designing techniques. Just 1 second. So the first technique is called rational protein design. Rational protein design approaches make protein sequence predictions that will fold to specific structures. These predicted sequences can then be validated experimentally through methods such as peptide synthesis, site-directed mutagenesis, or artificial gene synthesis. So what this means is, initially, computer-aided design is used, which predicts a certain sequence that will fold into the protein that we require. So you know there are softwares like AlphaFold, which received the Nobel Prize very recently, where if you enter a sequence, it can predict the three-dimensional structure of that sequence. So suppose you need a protein that has a alpha helical structure, a certain maybe combination of alpha, beta, alpha fold structure. So you can find out the sequence of amino acids that will fold into that kind of a structure using different software. Then you can carry out site-directed mutagenesis. So basically what that means is, using recombinant DNA technology, you can change the DNA in the plasmid to mutate. So we know the triplet coding, that 3 nucleotides will code for 1 amino acid. So by changing 3 nucleotides, we can change the 1 amino acid in the protein. So like this, we can carry out site-directed mutagenesis. So we have the original sequence. Now, the original sequence can be synthesized. Now, if we want to mutate residues in the original sequence and find out whether that increases or decreases the activity, that is site-directed mutagenesis. How that can be done is, basically the DNA we are inserting in the plasmid, we can change three nucleotides or more in the DNA to code for a different amino acid in specific regions of the sequence. Like that, one particular amino acid may be changed in one region, in another region, another amino acid will be changed, and so on. So site-directed, depending on the site, mutagenesis, amino acids are mutated. Then the plasmid is put in the expression system, generally E. coli, and the protein is allowed to be expressed. After expressed, the protein is purified by various methods we have seen before, and then the purified protein is tested. So you can see whether we can start with one initial protein, we can see its activity, and then by site-directed mutagenesis, we can optimize the activity. We can find out a sequence of amino acids which will have optimum activity. So this entire process is called rational protein design. Now, the advantage of this is that it is inexpensive and technically easy, since site-directed mutagenesis method is well developed. This entire recombinant DNA technology is well developed. But the drawback is that detailed structural knowledge of a protein is often unavailable. So for this to happen, you need to know what kind of 3D structure you want. So like I said, depending on what 3D structure of the protein we want, a sequence can be predicted. But how do we know what kind of 3D structure we want? For that, a very well-detailed information needs to be known about what structure of a protein has what function. Only if we know that, we can have a target structure of the protein, and then the computer program can predict what sequence will result in that structure. but often the structural knowledge is not available. And even if it is available, if we have structural knowledge from X-ray crystallography or even solid state NMR, we know the structure is a static picture of the protein. So we don't have an idea about the dynamics of the protein. So the lack of information about the structure and dynamics often causes a problem in this method. Now, there are some models. One second. Now we will look more into the details of this rational design. So protein function is heavily dependent on protein structure, and rational protein design uses this relationship to design function by designing proteins that have a target structure or fold. That is what I said in the previous slide, is that suppose we want a protein with a specific function, then we need to have a target structure or fold which will result in that function. So a detailed knowledge of structure function relationship is important. So by definition, in rational protein design, the target structure or ensemble of structures must be known beforehand. So that is the problem which I was discussing in the drawbacks of the rational protein design method, that we need to know the target structure. Very often it is based on the known structure of another protein which has similar function, but sometimes this knowledge is not there, and that is when this method cannot be used. In rational protein design, proteins can be redesigned from the sequence and structure of a known protein or completely designed from scratch in de novo protein design. So what this means, we can have a protein of known sequence and structure, and we can mutate certain amino acids in that protein to improve the function or get our desired function. Or we can start completely from scratch. So that is called de novo protein design. So there the entire sequence is designed from scratch. There is no prior sequence. One by one, the amino acid sequence is designed based on predicted structure. So de novo design takes a very long time. Comparatively, protein redesign is where most of the residues are maintained as the wild type and a few are allowed to mutate. So either we start from a known protein and by site-directed mutagenesis, that is protein redesign, the new protein can be synthesized, or by de novo design, which is designing the entire sequence from scratch. Now we will look at the energy function. So rational protein design techniques must be able to discriminate sequences that will be stable under the target fold from those that will prefer other low energy competing states. So suppose this is the folding funnel of folding energy landscape of the protein. We want a three-dimensional structure, which is here, suppose the native structure. But certain sequences may prefer some other structure, like the native structure is one of the possible structures, but suppose there is an even lower energy that is possible, that will be a low energy competing state. So that is not a good protein, because we want to design a protein which will without any doubt fold into the three-dimensional structure we want. If there are competing three-dimensional structures into which it can fold, then the protein has to be redesigned. So protein design requires accurate energy functions that can rank and score sequences by how they fold into the target structure. So that is a very important role of the energy function, that using the energy function, it can predict that suppose one sequence has a 98% chance of folding into the target structure and another protein has just a 42% chance. So obviously the 98% chance is the better design, because in 98% of the cases it will fold into the structure we want. However, the energy function must also consider the computational challenges behind protein design. So what this means is that if the energy function is too complicated, then even using very high processing computers to design a protein can take many months. So that is not like feasible with respect to time. So we need to come up with an energy function that is both accurate and simple for computational calculations. That is, both accuracy is maintained, but also it is not very slow. Molecular mechanics force fields, which have been used mostly in molecular dynamic simulations, are optimized for the simulation of single sequences. But protein design searches through many conformations of many sequences. So the molecular mechanics force fields, which we have studied in the previous class, that is okay for short amino acid sequence if you have only one sequence. But for protein design, you need to first optimize the sequence. So there can be multiple possible sequences. And within the sequence itself, there can be so many conformations, like the side chain chi1, chi2 can rotate, and so on. So there is much greater space within which the energy function has to search. So the energy function has to be appropriately designed. There are two kinds of main energy functions. One is the coarse-grained function, which is very fast computationally, but it does not go into the details. So it will give you an average view of the energy landscape, but it will not look into the details of each atomic interaction. So the advantage is that it is very quick to compute the lowest energy state. But the disadvantage is that sometimes there can be inaccuracies because it is not checking into the atomic details. The atomistic functions are much more accurate and sensitive to structure. So atomistic functions are one which considers interactions between each pair of atoms. So that is extremely accurate. But the problem is it is very slow computationally. It might take months to predict the structure of just one protein. So a balance has to be reached between these two. Now, what are some of the challenges in designing these energy functions? Water makes up most of the molecules surrounding proteins and is the main driver of protein structure. So modeling the interaction between water and protein is very important to protein design. Like suppose you have a protein here, the water molecules are shown, and you have certain hydrophobic groups in the protein shown in yellow. So whether the protein will prefer to fold itself and become globular such that the hydrophobic groups can interact with themselves and completely exclude the water molecules outside, that will depend on the protein sequence. If the protein has a lot of hydrophilic groups, then those groups will be exposed to the water. Basically, depending on the sequence of the protein, its three-dimensional structure will be determined based on the which is the lowest energy, and all interactions will be considered: protein-protein interactions, protein-water interactions, and so on. At any time, the number of water molecules that interact with a protein is very large, and each water molecules has so many degrees of freedom, and the water molecules can interact with other water molecules with the protein, and so it becomes a very complicated system. So most protein design programs, what they do, instead of modeling each water molecule separately, which is called explicit solvation model, they model the water as a continuum, which is called implicit solvation model. So they consider the water environment to be a constant medium having the solvation, polarization, and dielectric constant properties of water. So instead of individual water molecules, it is like the protein is in a homogeneous medium having the same property as the water molecules. However, the problem with this is that you can see in this example, there is one water molecule encapsulated inside the protein, which is stabilizing the structure of these polar groups. So sometimes individual water molecules like this can have a very crucial role in the structure of the protein. So failing to model these water molecules will not give you the true structure of the protein, and it will result in misprediction of the optimal sequence. So what is done to increase the accuracy is called microsolvation, where the few water molecules which are very important in the three-dimensional structure of the protein are modeled separately near the polar groups of the protein, and the rest of the water molecules is considered as a continuum So basically something between the explicit and implicit solvation models is used, and it's called microsolvation. Another problem is that the number of candidate protein sequences grows exponentially with the number of protein residues. For example, if you have a protein of 100 amino acids, there are 20 to the power 100 possible sequences. Additionally, each amino acid side chain can have multiple conformers. So this results in an exponential number of conformers for each sequence. So all this becomes very difficult to predict. And if the protein has to sample all these conformations, then the folding time will be of the protein will be so high that it is not feasible. A designer is only interested in the best sequence. So the protein design algorithm only requires the lowest energy conformation of the lowest energy sequence. In these cases, the amino acid identity of each rotamer can be ignored, and all rotamers belonging to different amino acids can be treated the same. Basically, to simplify the problem, we don't consider each amino acid separately, because if we do that, it results in 20 to the power 100 sequences or more. Rather, what is done is that all amino acids are considered similar, and only the lowest energy of the total sequence is considered. So the energy function is something like this. We have let Ri be a rotamer at residue position I in the protein chain, and Eri is the potential energy between the atoms of that rotamer. To this, you have, and so Ri is a certain rotamer at residue position I, and Ei is the energy of that rotamer. Now Ri is interacting with a rotamer at position J, that is Rj. So there is an inter-residue energy Eij, and this is added up for all the residues. So suppose this is residue 1 and you have 5 residues. So interaction of 1 with 2, 1 with 3, 1 with 4, 1 with 5, all of that is added up. So you have the energy of each, and this is added up for all residues. So this is the sum of the energy of each residue within itself and the inter-residue energy of each residue with the other residues. This is added up for all the residues, and this term is minimized. So the computer program basically computes this energy term for all possible conformers, and wherever is the this is minimized, that is the lowest energy information. But of course, there are ways to speed up the process. Like the computer does not have to sample all possible conformations. We'll discuss that in the next slide, that how this process of checking for the minimum energy conformation can be speeded up. So there are several algorithms that have been designed for this problem. There are certain algorithms called exact algorithm, like dead end elimination or DEE. So what this algorithm does is that, suppose there is a rotamer IR, so it is checking the energy of the rotamer IR, and it is checking the inter-residue energy of IR with the other residues. So suppose a particular rotamer, and you have the rotamer IT. So which rotamer that particular residue will, given a residue I, it can have different rotamers. Let us look at two rotamers. One is IR, another is IT. So if the minimum energy of the interaction of residue rotamer IR with the other residues is greater than the maximum energy of the rotamer IT, then the previous rotamer is eliminated. So what this means is that suppose one amino acid can have so many rotamers. So how we need a procedure by which many of the high energy rotamers are eliminated. So you can see we don't need to scan through the entire we don't need to scan the entire energy landscape. We can just scan the low energy regions. So if we scan these regions, that should be enough. So how this is done is that the algorithm will quickly check, and all the high energy rotamers it will eliminate. So that is what I was saying, is that it will check the rotamer energies, and if the minimum energy of one rotamer is greater than the maximum energy of another rotamer, it will eliminate the greater energy rotamer. So you will only be left with a set of rotamers of low energy, and then they can be quickly put in the energy function, and the global energy minima can be found. So this is known as an exact algorithm or dead end elimination. So it guarantees that the solution is like you will reach an accurate solution. But the problem is that it takes a long time, because ultimately it still has to put the energy of each rotamer in this equation in order to eliminate the high energy rotamers. So still many rotamers have to be sampled to narrow down the search area. So there is no guarantee about the runtime. So that is one problem of this exact algorithm. There are other algorithms which are faster than this, like the Monte Carlo algorithm, where what happens in the energy landscape, you start from any region and you choose, you can either go forward or backward depending on the probability. If the probability of say I start from here, and the probability of going from here to here is 1, so definitely it will go here. Again, the probability of going from here to here is 1, so definitely it will go here. But the probability of going forward is very low, 0.07. So this path will not be taken, it is shown in red. Like that, by jumping from one confirmation to another, the Monte Carlo algorithm gets to the lowest energy confirmation. But the problem of this is that often the molecule can get stuck in a local minima. So suppose you have one minima here, which is close to the global minima, but not. So this is the global minima, and you have something here. So when your molecule is here, probability of going here is also low, probability of going here is also low, because this is a low energy state. It will not want to escape the low energy state. So then it will give a wrong prediction. It will predict that this is your native structure, but this is not your native structure. This is your native structure. So the problem with algorithms like the Monte Carlo algorithm, which is kind of a random sampling based on probability, there is no guarantee on the optimality of the results. So sometimes it can mispredict a local minima as the global minima. So these are some algorithms which have been used to make the process of finding the lowest energy state faster. And the more advanced algorithms uses a mixture of different algorithms. We don't need to go into the details of that. So as I was saying, 3 nucleotides will code for 1 amino acid. So there are different kinds of mutations that can happen. One is called a substitution mutation, where one nucleotide is changed into another. So A can be changed into C, and so on. Another can be, so I will give some examples here. So suppose you have TTC and that gets mutated to TTT. This is a substitution mutation where C is converted to T. But interestingly, TTC, so this is T in DNA, in RNA it will be U. So UUC is coding for phenylalanine. So this will code for phenylalanine. This is coding for phenylalanine. Interestingly, TTT also codes for phenylalanine. So you can see both UUU and UUC code for phenylalanine. So this is called a silent mutation, because even though there is a mutation, there is no effect because it is resulting in the same amino acid sequence. Similarly, if AAG There is a mutation to AAA. Again, substitution mutation, G is mutated to A, but both are coding for lysine. So this is a silent mutation. Now there can be missense mutation. Like suppose you have initial sequence was AAA, now you have AAT. So instead of phenylalanine, leucine is inserted. Then the amino acid sequence itself becomes different, and it is called missense. So you can see, because of this mutation of AAA becomes AAT, that results, instead of phenylalanine you have leucine, and this is a missense mutation. A nonsense mutation is where suddenly the amino acid sequence stops. So instead of ACC, the middle C is mutated to T, so it becomes ATC. And ATC is a stop codon. That means whenever this codon is encountered, the translation will stop. So this is called nonsense, because the translation stops at that point. Now, another kind of mutation can be the insertion mutation, where a nucleotide is inserted. So here you can see it was TAT. Now, before this T here, an A is inserted. So because of that, what will happen? Here it was TAT, CGG, so on. Now, because an A is inserted, the codons become different. It becomes ATT. This T is now moved here, so it becomes TCG, and so on. So the amino acids also become different. Instead of isoleucine, alanine, asparagine, it becomes tyrosine, serine, glutamine, because the entire codon is moved by one nucleotide, and so the codons code for different amino acids. So this is addition or insertion, where one nucleotide is added, and it is frame shift because the reading of the codon completely shifts. There can also be frame shift by deletion, where multiple nucleotides can be deleted in the sequence, and that will also result in a completely different sequence, or even the sequence can suddenly stop. So these are different kinds of point mutations. Now we will look at directed mutagenesis. So the hypothesis in directed mutagenesis, one second. The hypothesis in directed mutagenesis is that natural sequence can be modified to improve a certain function of the protein. The sequence must not change too much, otherwise the structure itself will be disrupted and folding will not happen. So the new sequence is similar to the native sequence, with mutations at only a few places. That is point mutations. So one can be side-directed mutagenesis. So we know that a particular region is the interacting region, and we carry out point mutations in that known area. So we will get, one is the wild type DNA and one mutated DNA, where mutations have been done at regions where we know interaction happens. So that is side-directed mutagenesis. Another is random mutations, where mutations can happen in all regions within the DNA. So we will get an entire library having the wild type DNA as well as mutated DNA. So in random mutagenesis, mutation happens in random regions of the DNA. So you will have an entire library of wild type and mutated DNA. And then these DNA will be put into plasmids, and recombinant DNA will be there, and the protein corresponding to those DNA will be expressed. So we will get an entire library of wild type protein as well as mutated proteins, and these can be used to check the biological activity. And using these techniques, we can find out that which mutations cause an improvement in biological activity, which mutations completely change the biological activity, and so on. So another technique is the de novo protein engineering, which is there is no prior experimental information. The protein is completely designed from scratch with the help of protein structure prediction algorithms. So how that is done is that you you know a certain region is the hotspot region of the protein. So we can basically put like, suppose we know that there is a region of 40 amino acids where the interaction is happening. So in that 40 amino acids, there can be many sequences possible, and each of these sequences can have a different three-dimensional structure. So all these 40, say, let us consider say 5,000 possible sequences will have different three-dimensional structures that can be predicted by computational programs. Then the DNA sequences corresponding to these peptides can be synthesized and replicated in plasmids, and all these can be synthesized, purified, and then used to study. So this is basically de novo protein design, where you are designing the protein from scratch. You know nothing about the protein. The only thing you know is which region is the interaction happening. Otherwise, there is no prior protein from which you are taking a reference or target structure. You are completely designing the protein from scratch. So this is considerably harder than conventional protein engineering based on rational design, but there are certain advantages. So in rational design, basically, we were taking a target structure and modifying different residues to reach a structure close to the target structure. But the problem is that in natural proteins, we cannot change the sequence too much without disrupting the structure. Compared to that, if we design a protein from scratch, there is much more flexibility in the structure allowed. Another thing, we can design enzymes with completely new activities which did not exist in nature. Whereas if we were building on a previously known structure, maximum we can improve on previously existing activities. But by this de novo design, we can completely design a new protein altogether. Of course, a huge amount of computational time is needed because you have to start from scratch. But slowly, with computational advantages, this is becoming more and more possible. So one second. So there are certain methods which predict protein structure. One is knowledge-based methods, where a protein fold recognition technique involves incrementally replacing the sequence of a known protein structure with a query sequence of unknown structure. So basically, suppose you have an unknown sequence, which you have a certain sequence which you don't know what is the structure of that protein. So that sequence is compared with other known sequences for which the structure is known. So suppose we have a sequence X, Y, so on, Z. So we know from here to here, we compared it with known sequences and find out it has a helical structure. Then from here to here, we compare with known sequences and find out it has a beta sheath structure. So like that, the sequence is compared with other known sequences, and it is kind of overlap to reconstruct the possible structure of this new sequence. So that is the method based on knowledge. Another is the ab initio method, where the basic idea is to build a function that simulates real physical forces and potentials of intermolecular interactions. So we will have a function which will be able to predict the different interactions between the different amino acids, and by energy minimization and molecular dynamics, eventually it will reach the lowest energy structure, that is the predicted structure. So disadvantage is that computationally it can only be applied to short sequences. Otherwise, it becomes too complex for the computer to handle it. So that is the ab initio method, where starting from a completely unfolded sequence, you can reach, by using energy minimization and molecular dynamics, you can reach the predicted lowest energy sequence. There are certain software methods available for this structure prediction. One of them is called ITASR, which stands for Iterative Threading Assembly Refinement Structure Prediction. So there are certain steps in which this software predicts the structure. Step one is called threading, which refers to a bioinformatics procedure for identifying template proteins from a solved structure database which have a similar structure or similar structural motif as the query sequence. So this kind of starts like the knowledge-based methods. So you have a database where you have known sequence and structure of several proteins. So the first step is to identify proteins which have a similar sequence or a similar structure compared to the protein that we are looking at. The second thing is structural assembly. So continuous fragments in threading alignments are So like I was saying here, the sequences are put together to predict the structure. This is basically what this software is doing. So it is first aligning known sequences with the new sequence, and it is taking fragments of the known sequences which are matching to the new sequence. So if that fragment of the known sequence is predicting a certain secondary structure, it is taking that, it is then joining that to the next part of the sequence which is predicting some other structure, and they are assembled into an initial model. So that is structural assembly, where from all the known sequences, you have certain secondary structures which are predicted. All these secondary structures are put together to predict a structure for the unknown protein. Then within the structures predicted, it is refined even more. So some of these structures may be not allowed by the Ramachandran plot. They may be energetically very high. So like that, a refinement is done. Based on that, you get a, so you can see here there was a lot of like unpredictability in the structure. You can see uncertainty is there. This you can see the places which are shown in very light color here are the uncertainties. So as you refine the structure, the uncertainty is lost and you get a general backbone of the structure. And the function of the final step is structure-based functional annotation. So whatever structure you are getting after refinement, that is compared with the structure of known proteins in the database. So that known protein has a particular function. If the structure matches the structure, so suppose the structure you're getting from the unknown protein is a combination of structures of 3 known proteins. So whatever is the structure of those 3 proteins, the unknown protein will have a combination of those functions. So like that, by structural and functional relationship, that is the final step, where the function is predicted based on the known structure function of the database. So this entire process is done by a software called iTassert, which is, and it is iterative. So this refinement step can be done multiple times to get a better and better model. And this, of course, depends on how good the database is. So if the database is good enough, then the final structure prediction and function prediction will be very accurate. That this was iTassert, which is a one kind of structure prediction server. Another is the Rosetta structure prediction. So here what happens is that this software includes different algorithms for computational modeling and analysis of protein structures. A suite of modeling and search tools is used which can calculate the energy of a folded protein and search for the lowest energy sequence of a given structure or lowest energy structure for a given sequence. So suppose you have a certain sequence. First, it will calculate the possible 3-dimensional structures, and it will find which of these structures has the lowest energy. First, a coarse-grained energy function can be used to narrow down the possible structure, and then an all-atom or atomistic energy function will be used to get a more accurate idea of the lowest energy structure. Eventually, through different energy functions, we will reach one structure, which is the lowest energy possible structure of this sequence. Or if we are starting with a structure, then we will find the lowest energy sequence possible for that structure. So the Rosetta method is based on minimizing the energy. We saw the Monte Carlo algorithm where from high energy, it can go to low energy by randomly going through the energy landscape in a probability based method. So using the Monte Carlo algorithm, the Rosetta software basically predicts the lowest energy structure of a given sequence. Or if we start with the structure, it will predict the lowest energy sequence of the given structure. So using this, again, protein can be designed based on a particular sequence, or if we have the sequence, we can predict the particular structure. There are certain applications of protein design. One is the DNA origami. So DNA origami is the nanoscale folding of DNA to create arbitrary 2 and 3 dimensional shapes at the nanoscale. So we know that DNA, there is the concept of complementary base pairs. That is, A will pair only with T and G will pair only with C. So because of this principle of complementary base pairs, we can design DNA in such a way that we can build different molecules using the DNA. So imagine this is a DNA strand. And here we have complementary base pairs. So this region of the strand will come close. And if here the base pairs are not complementary, this region of the strand will stay far from each other. And like that, we can build different two-dimensional structures. This really doesn't have, like many of these structures don't really have a use, but they have been built to demonstrate that we can design DNA in such a way that it will fold into different kinds of structures. Using this particular concept, Chen wanted to identify hydrogen bonding strategies that will allow him to design perfectly orthogonal protein pairs. Like DNA, A interacting with T and C interacting with G, if we can identify pairs of proteins that will interact only with each other and not with similar proteins, then that will help us to build any kind of 3D structure based on these orthogonal protein pairs. So that from there came the concept of blob level protein design, where basically protein units of designed function can be put together to engineer a fusion protein with a completely new function. So, and that can be done if we have these kind of orthogonal protein pairs, which can be paired up with each other in a stable manner, and the net fusion protein will have a completely new function, or it can sometimes have the functions of both the proteins and so on. Now, about directed evolution. Just 1 second. So now we will discuss the concept of directed evolution. Natural evolution of enzymes has existed since the emergence of life on Earth. Genes have mutated and proteins have evolved to improve the fitness of an organism to tackle conditions in new environments. Directed evolution of enzymes and binding proteins is a man-made process built on molecular insights, which moves the evolution process into the laboratory and speeds it up. Basically, directed evolution is an iterative process which involves the identification of a starting state protein, diversification of its gene. So we start with a certain protein that is expressed by a wild type gene. We diversify the gene by mutagenesis. Then the gene is expressed in an expression system like E. coli, and the PCR can be used to amplify the DNA. It can be put in the plasmid and expressed in E. coli to get that particular mutated protein. And that protein can be screened for different functions. So suppose some of the mutated proteins match the desired function, then it will be a successfully mutated protein. Otherwise, the process will be repeated with different mutations till we get, so the diversification will be repeated, screening will be repeated till we reach a satisfactory performance level in terms of the protein activity. It can be enzyme activity, binding affinity, or specificity. So this is directed evolution. Sometimes if we want even better results, we can take this resultant mutated protein and mutate it even further. So the process is iterative. You can repeat it as many times as you want till you reach, get a protein with the desired level of function. So this directed evolution of enzyme tailors them to operate in new reaction conditions, optimizes catalytic activity towards new substrates, and make them catalyze new chemical reactions also. I will be discussing these applications in the coming slides. Now, while designing these mutated proteins, there are some insights which help. It is not possible to randomize every position in an enzyme. The typical size of an enzyme is 200 to 300 amino acid residues. And we know if we randomly mutate each position, it will be a huge number of sequences which cannot be studied. So we have a database which makes it clear that mutations near the active site and some mutations slightly away from the active site will contribute to the catalytic activity. So this knowledge is very important. That so that was what we studied as site-directed mutagenesis, where we know that which region needs to be mutated. Stemmer introduced a DNA recombination strategy termed DNA shuffling to the evolution of enzymes. This was an efficient way to propagate beneficial mutations while increasing the size of a DNA library through random fragmentation and reassembly of genes. So what is this DNA shuffling? The first is that random mutation is introduced in the gene for the enzyme that will be changed. So suppose this DNA is coding for the wild type enzyme. Random mutations are introduced in the DNA. The genes are inserted in bacteria which use them as templates and produce randomly mutated enzymes. So these DNA are put into plasmids and they are expressed and we get many mutated enzymes. The changed enzymes are tested. Those that are most efficient at catalyzing the desired chemical reaction are selected. So these enzymes are all tested for the biological activity. Those which are very efficient, they are isolated, and the ones which are not working or less efficient, they are discarded. Then in the gene of the selected enzyme, again the mutation process begins. So again, this is an iterative process, which is basically from one round of the process, you are getting an idea that which mutations are affecting the activity positively. So that will give you a knowledge about the next set of mutations. That is again which interaction. So that will help in narrowing down the number of mutations required to improve an enzyme. Now we are selecting. So from these mutated enzymes, what are we mutating them for? They are mutated for several different properties, one of which is thermal stability. So directed evolution of enzymes can be used with the aim to increase the thermal stability. So here the assay that is used to find out which enzymes are good, for that a heat treatment and activity assay can be used. That is, you are treating each of these enzymes with heat and finding out if they are retaining that activity or they are getting denatured. So that will help in isolating more and more thermally stable enzymes. Another can be to improve or change the catalytic activity. So suppose the wild type enzyme is catalyzing for a certain reaction that is called the natural reaction. We can design an enzyme by the directed evolution of enzymes to catalyze for a completely new reaction. This is possible as long as the natural reaction and the new reaction have some overlap in enzyme. That is an enzyme that suppose an enzyme that is catalyzing this much of the natural reaction is also catalyzing small amount of the new reaction. So certain level of enzyme promiscuity. Certain level of enzyme promiscuity is essential. That is, some low level of activity for the intended reaction must be there. So then mutations can be carried out to reduce the level of activity for the natural reaction and increase the level of activity for the new reaction. And by multiple processes of this DNA shuffling, we will ultimately make a new enzyme which will catalyze a completely new reaction. Then we can look at different metabolic pathways. A strength of the directed evolution methodology is the ability to co-evolve enzymes in biosynthetic pathways. In one example, Arnold and co-workers evolved a multi-enzyme pathway for carotenoid production in E. coli. So for carotenoid production in E. coli, they found that multiple enzymes could catalyze this biosynthetic pathway. So while designing, suppose this kind of improving catalytic activity, they must have seen that many mutations were giving the same results and in the carotenoid production. So they found that multiple enzymes they found which can parallelly catalyze the this production in this biosynthetic path. Another application is in biofuels. So a leading candidate of biofuel is 2-methyl-propanol or isobutanol, which can be produced using a biosynthetic pathway in recombinant E. coli. However, the problem is that two enzymes in the pathway required NADPH as a cofactor, but the normal metabolism during growth of E. coli produces NADH. To resolve this obstacle, directed evolution was used to alter the cofactor dependence of the enzyme. So instead of NADPH, they can rely on NADH. This makes the enzyme and organism more suitable for biofuel production. So what was happening is that while producing isobutanol, the entire pathway of the production, it required NADPH, but the much more widely available cofactor was NADH in E. coli. So by directing the evolution of E. coli, directing the evolution of these enzymes by DNA shuffling, slowly mutations were done in the DNA so that the enzyme produced will use the cofactor NADPH instead of NADH. That way, so that the directed evolution was done by DNA shuffling, so that the new mutated enzyme produced depends on NADH. So what that will do is that NADH is available in large quantities to E. coli. So now these enzymes, which are in the pathway of producing isobutanol, they will be much more effective. So the efficiency of isobutanol production is increased greatly by mutating the enzyme such that they depend on the cofactored NADH, which is more widely available than the cofactored NADPH. Just give me 1 second. Other applications of this site-directed evolution. One is catalyzing the new chemical bonds. So carbon-silicon bonds are common in human-made chemicals, but they are absent in biology. Nature has not evolved enzymes that catalyze the formation of carbon-silicon bonds. But directed evolution can be used as a strategy to ensure that such chemistry invented by humans can also be conducted by help of enzymes. So what is done is we start from the enzyme cytochrome C from Rhodothermus marinus, And from this enzyme, directed evolution is used to create another enzyme that catalyzes silicon carbon bond formation 40 times better than cytochrome C. So now you have a new enzyme, the evolved cytochrome C, which is catalyzing carbon silicon bond formation very fast. So even for human-made reactions, new enzymes that catalyze those reactions can be developed by directed evolution. Another application is enantioselectivity of enzymes. So directed evolution is an efficient way to improve the enantioselectivity of enzymes, that is enhancing their performance in asymmetric catalysis. So we have seen in directed evolution, basically you can have random mutations in the DNA that can be expressed to get a library of proteins. Then in that library of proteins, you can carry out the chemical reaction and see whether that reaction is that is, the enzyme will react with one particular, suppose it is reacting with the R enantiomer, it will not react with the S enantiomer at all. So there is selectivity between enantiomers as well, and that is called asymmetric catalysis. So from an enzyme that is not selective to enantiomers, that is, it reacts with the R and S equally, by directed evolution, a new enzyme can be created which is selective, which selectively interacts either with R or the S enantiomer. And so they can be used in the production of chiral substances with high enantiomeric purity. So suppose it is interacting only with R enantiomer, then it will result in the product also having R, the product also which has the R chirality. So you will be able to produce chiral substances with very high purity because the enzyme will be interacting with only one of the enantiomers. Using this kind of directed evolution on transaminases, S selectivity in the conversion of the ketone beta tetralone to the corresponding amine was greatly enhanced. Previously, S enantiomer was formed 65%, while the rest was R. But after the evolution of this enzyme, the selectivity became 94%. So 94% of the product was S enantiomer. So there was great enantioselectivity. So that can also be improved by directed evolution. Now we will look at machine learning. Machine learning assisted protein engineering. Just one second. So machine learning assisted protein engineering. So the bottleneck in the entire process of directed evolution is the screening step, where we have to check the activity of all the proteins. And that requires a lot of experimental efforts. To reduce the experimental effort associated with the directed protein evolution, and to explore the sequence space encoded by mutating multiple positions simultaneously, machine learning was incorporated in the directed evolution process. So what happens here is that molecular dynamic simulations, which predict dynamic structural changes for protein variants, have been used to predict changes in structure and protein properties caused by mutations. However, full simulations are also resource intensive, requiring hundreds of processor hours for each variant. So a mechanistic understanding of the reaction and ideally a reference protein structure is important to have. So what this is saying is basically, if we have to experimentally test the activity of each of the mutated proteins, it requires very long. Rather than that, if we use something like molecular dynamic simulations, where previously molecular dynamics was predicting how the protein will move with time, but if we can use similar kind of computation to predict that how the structure of the protein will change with time, and depending on the structure, how the function will change, then we don't have to screen all the mutants. We can only screen those mutants which have the structure which relates to the function that we want. So basically, machine learning uses a set of algorithms that make decision based on data. So suppose from mutation, we get a variety of sequences and having different structures. So you have the data about the structures, and based on the structures, machine learning can make a decision because it has the idea and database of the known structure function. So the computer has the knowledge of the known structure and function of known proteins. So now from the mutated proteins, it can look at the structure of the mutated proteins and eliminate the structure which does not have the desired function. So those, it is basically narrowing down the number of experiments you have to do. Instead of doing experiments on say 50 mutants, it is screening out 40 mutants because they don't have the structure which will result in the function we want. So based on known data, machine learning is taking that decision to narrow down the number of possible experiments that has to be done. So it enables rapid prediction of new variants, whereas if we use only experiment, it would take too long. Similarly, if we use only theory, it would take too long. That is what they are saying, that only simulation is also too long and will take too many processor hours. So a kind of simulation combined with experiment is done to optimize the time required for this directed evolution process. So in this machine learning, the concept of model development is this, that we have quantitative structured activity relationships to evaluate different set of structures that we want. So what is known is we have a database of proteins which have a certain structure and a certain function. So there is knowledge about what kind of structure results in what kind of function. So what was established in machine learning is sequence activity modeling, that is QSAM. So this sequence modeling activity was established to elucidate features that are amino acid descriptors for detecting beneficial amino acid patterns and protein variants inside a defined sequence space. So from the database of known structured activity relationships of protein, the machine learning algorithm will go through the known database and find out sequence activity relationship. That is, which sequence of amino acids will result in the desired structure. That is, which sequence will result in the desired function. So so the algorithm basically fits a linear function based on sequence activity relationship to classify substitution. So instead of randomly doing mutations, the algorithm will predict that which mutations will result in a beneficial mutated protein and which will result in a detrimental mutated protein. So all mutations don't need to be checked. Based on the machine learning, the algorithm will predict that which is the sequence mutations which will result in the function that we want. So we identify the beneficial substitutions and the detrimental substitutions from known database of proteins. And these identified substitutions can be introduced or deleted for the next round of evolution. This has improved the catalytic function by 4,000-fold. In a target enzyme, it was seen that just by 35 substitutions, 35 mutations, the catalytic activity was increased by 4,000 times when machine learning was included in the directed evolution process. So basically what machine learning is doing, it is going through the database of known structured activity relationships and it is finding out sequence activity relationship. So it is predicting what sequence of amino acid will result in a certain function. Based on that, it is doing the mutation. So instead of random mutagenesis, it is identifying what mutations will be beneficial to the activity that we want. So instead of randomly getting a series of mutants, we are getting mutants which are actually doing the function that we want and improving that function. So based on that, instead of the directed evolution based completely on experiment, this is narrowing down the number of mutations required. And you can see an example here. Just by 35 mutations, the catalytic function is improved 4,000 times. One problem, however, is that mutations can have a propensity that of epistasis. So what is this epistasis? Epistasis is a phenomenon in genetics in which the effect of a gene mutation is dependent on the presence or absence of mutation in one or more other genes, which are called modifier genes. So epistatic mutations have different effects on their own rather than when they occur together. What this means, suppose we have green fluorescent protein, the wild type is in the middle. We have carried out multiple, so this first circle is the first round of mutations. Among these mutations, 1, 2, 3, 4, 5, and 6 of the mutant proteins are showing the green fluorescent activity, but 2 are not showing any activity. Now, 2 mutations can be combined. When 2 mutations are combined, you can either get, you can see one which is showing the fluorescent activity, one is not showing. When these are combined, they are cancelling out and the result is not showing any mutation activity. So this is called negative epistasis, where accumulating mutations can cancel each other out. So this is showing fluorescence, this is not showing fluorescence. When both mutations are there together, there is no fluorescence. Sometimes it can lead to positive epistasis also, like this is showing fluorescence, this is showing fluorescence. If you add both mutations, you might have greater fluorescence. So that will be positive epistasis. So basically the effect of different mutations on one another, so that is what epistasis is, where the effect of a gene mutation is dependent on the presence or absence of mutation in other genes. So suppose here you have a fluorescent mutant, here you have a fluorescent mutant. If they are combined, this is showing fluorescence. So here there is no epistasis. But here 2 mutations are combining to give a variant which is not fluorescent, that is negative epistasis, where the activity is cancelling out. Now here that example is not there, but suppose you have an example where 2 mutations are coming together. Suppose these 2 mutations are coming together to give a more highly fluorescent type of protein, then it will be a positive epistasis. So that is one problem in this kind of multiple mutations, but machine learning is a useful method to find functions which can predict this behavior. There is a set of programs called the Iterative Protein Redesign and Optimization Suite of Programs, or IPRO. Iterative Protein Redesign and Optimization Program. It redesigns proteins to increase or give specificity to native or novel substrates or cofactors. This is done by repeatedly randomly perturbing the structure of proteins around specified design positions to identify the lowest energy combination of rotamers and determining whether the new design has a lower binding energy than prior ones. So suppose you have different conformations of the proteins. Basically, you are changing the, you are mutating the protein to find out different 3D structures which have different binding energies with a particular ligand. And you are, and then that is the competing ligands. But we want specific binding with a particular ligand only, that is the targeted ligand. So we can perturb the structure of the protein around a particular design position, meaning we know that which region of the protein is interacting with this blue ligand. We can perturb that region so that it no longer interacts with the blue ligand and instead it interacts with our target ligand, that is this green ligand. So we can perturb the structure by different mutations, and it has to be ensured that the binding energy of the target ligand is significantly lower than the binding energy of all the other competing ligands. Only then we can ensure that the mutated protein is specific to the targeted ligand and does not bind with any other competing ligands. So this is a program which will redesign the protein till it optimizes the mutated protein so that it has the highest binding affinity and specificity to the target ligand. EPRO also provides an integrated environment for grafting a binding pocket into an existing protein scaffold. So suppose you have a stable three-dimensional structure of a protein, you can include a binding pocket in that stable three-dimensional structure. It can also predict an antibody's tertiary structure based on sequence. It can enhance enzymatic activity, which was shown here. The activity for the target ligand was enhanced. Assess the structure and binding energies for a specific mutant. So it will give you the structure and binding energies of different mutants. One of the programs of EPRO is also called Pore Designer, was successfully developed to redesign bacterial channel proteins. So channel proteins basically are found in bacterial membranes, which allow substances to enter or leave the cell. So initially the protein had pores of 1 nanometer size, but the protein can be redesigned in any way to reduce or increase the pore size. One mutation shown here is where the aspartic acid and serine in the pore were both mutated to tryptophan. And this reduced the size of the pore because tryptophan has larger side chain. So the size of this pore was reduced. And also it was more hydrophobic because tryptophan side chain is hydrophobic compared to aspartic or serine. So the pore became more hydrophobic. So it is more selective to hydrophobic particles passing in and out of the cell. So this mutation was predicted by this EPRO set of programs, and it helped in changing the pore size as well as making the pore more selective to hydrophobic substances. Another success story of protein engineering, basically computer-aided design has been used to engineer complex properties of a highly ordered nanoprotein assembly. A protein cage, E. coli bacterioferritin. So E. coli bacterioferritin is a protein which does not have any natural self-assembly, because whenever self-assembly is happening, there is a gap. You can see this is formed by self-assembly of this E. coli bacterioferritin, but within this self-assembly, there is a gap where there are water molecules. This was found by computational analysis and comparison to homologs, that at the interface of self-assembly, there is an existence of an interfacial water pocket, which is connected to two asparagines. Because of this water pocket, there is not much interaction between the monomers, and so it is not a very stable aggregate. If these two asparagines are replaced by hydrophobic amino acids, so that the water pocket will be eliminated and the hydrophobic amino acids will interact by hydrophobic interactions and hold the self-assembly structure together, it was found that the stability of the cage formation increases if these asparagines were replaced by leucine. You can see if the asparagine is replaced by leucine, cage formation, this self-assembly formation is very much stabilized. But if it is replaced by phenylalanine or tryptophan, it is not that much stable. So like that, the protein was engineered in such a way that the asparagine was removed. This eliminated the water pocket because the waters were held by the asparagine, and the asparagine was replaced by leucine. So because the leucine side chains are aliphatic and they can have stabilizing hydrophobic interactions, because of this mutation, the stability of the self-assembled structure was improved. And so now this can be used as a kind of vessel to carry some substance inside or outside the cell. That is what self-assembled structures are used for. But this is one success of protein engineering, where the native structure was not self-assembling well, but just one mutation caused a great increase in the stability of this self-assembled structure. Other things that protein designing can be used for: one is building better vaccines for pathogens. Another is expanding beyond nature's alphabet of 20 amino acids. So we know the 20 naturally occurring amino acids, but it can be expanded beyond that by protein engineering. Advanced protein containers can be created for targeted drug delivery. So this kind of self-assembled structure can be used as a protein container for targeted drug delivery. Smart protein systems can be designed that can identify cancerous cells and specifically target cancerous cells and avoid the healthy cells. And next generation nanomaterials can be created for use in solar energy capture and nanofabrication. So we won't go into the details of these, but you can just remember them as different applications for protein engineering. Now, some of the advances of protein engineering we will talk about. So this ultra potent vaccines. Researchers from IPD created novel immunogens against the respiratory virus RSV, which was in COVID-19. So this ultra potent COVID-19 vaccine was designed completely by protein engineering. It combined, so we talked about fusion proteins, how the characteristic functions of two or more proteins can be combined by fusion. So that was done to get this ultra potent vaccine, and animal testing has shown that the activity is 10 times greater than mRNA vaccines. So this was completely designed by protein engineering. Another advancement in this field is the cancer immunotherapy. So in 2019, IPD created compact proteins that stimulate the same receptors as IL-2, a powerful immunotherapeutic drug, while avoiding unwanted off-target receptor interactions. So basically, what was happening is that the T cells of the immune response were binding with the tumor and getting deactivated. This immunotherapy drug is blocking the tumor cells and preventing the deactivation of the T cells, so that the patient does not become immunocompromised. So not this protein was designed by protein engineering to be similarly effective as IL-2, which was already a good drug, but additionally it was made very specific. So if you remember, we just talked here how specificity can be increased by different set of programs which do protein engineering. So this cancer immunotherapy drug was made extremely specific so that it targeted only the cancerous cells. Then rapid antivirals have also been designed by this technique. In 2011, IPD designed synthetic mini proteins that target conserved surfaces on influenza hemagglutinin from H1N1 pandemic flu virus. So this mini protein again has been completely designed by protein engineering. It was a de novo protein that was designed from scratch, and it has shown very good effects. Even and they have also shown that this protein can be used as diagnostic reagent in low cost diagnostic test strip to provide improved performance on influenza assay compared to a traditional antibody based capture system. So the diagnostic test traditionally used to take a long time based on antibody captured system from the blood drawn from the patient. But this protein can just be put on a diagnostic strip, and it is a very quick way of testing whether the patient has this disease or not. Another advancement has been in immune silencing. So in 2014, IPD scientists were the first to demonstrate that computational design can be used to eliminate T cell epitopes from proteins by altering the amino acid sequence without affecting structure or function. So it was completely designed computationally, and they had shown that computer software has advanced so much that you can completely design a protein computationally, and it was successful like the native protein and had the same structure and function similar to the native protein. And this immune silencing approach has been used to treat cancer with very good activity and anti-tumor activity. Another application has been in heme protein. Now, heme protein is viewed as a self-assembling, fully DNA encoded catalyst whose electronic and steric properties can be tuned by evolution. It is an iron complex that can be modified simply by mutation of the DNA and make it capable of catalyzing very interesting reactions. So from biology, several reactions are natural to the heme protein. One is conversion of the CH bond to COH bond or C double bond C to epoxide and so on. So there are a set of natural reactions which are catalyzed by the native heme protein. Enzyme catalysis can be done to create mutated proteins from the natural heme protein that catalyzes a completely new set of non-natural reactions and opens up a whole world of possibility. So all these are non-natural interactions where C double bond C is converted to cyclopropane, carbene XH insertion, and so on. So all these are non-natural reactions using synthetic reagents, and they can be catalyzed by mutated heme proteins prepared by directed evolution. So if you remember, we talked few slides before, we talked about carbon silicon bond, how there are no natural enzymes to catalyze the formation of carbon silicon bonds, but non-natural enzymes created by mutation of the natural enzymes can do that. So this is a similar way in which the heme protein can be mutated by directed evolution to catalyze an entire set of non-natural reactions. There are also new approaches and future directions to this protein engineering field. Basically, we'll talk about some of the future directions in which the protein engineering field can go. Cadet et al. Came up with a supervised learning of enantioselective enzyme sequences and activity of n individual point mutations to predict the activity of all combination of these point mutation. What this means is that you have an amino acid sequence and the sequence is given a numerical code. So you can see each amino acid is defined by a certain numerical code which can be Fourier transformed to define a protein sequence. Now, every single mutant of this amino acid sequence can similarly be coded and Fourier transformed to show this kind of a spectrum. And from this kind of coding, the protein activity can also be designated a certain number. So basically the thing is that you are numerically encoding the sequences, both the wild type and the mutants, and the experimental activity of the observed sequences. Just one second. So you are numerically encoding the sequence and the experimental activity, and there is a method to predict the activity by combining the mutations. So we don't need to go into the numerical details of this, but basically Cadet et al. Has come up with a way by which protein activity can be predicted. So basically we have a set of mutants and a set of the activity of the mutants. Now the computer has been, you can imagine it like AI. So AI learns as it goes. So as it goes, the program learns about the relation between the sequence and the activity. And it is able to predict of a completely new protein which combines different mutations, it can predict the activity moderately correctly. The correlation between the predicted and experimental activity has been found to be 0.81, which is not a perfect model, but this is a new direction in which this field is going, where it can be from the knowledge of different point mutations and their effect on the activity, the uh, predict the activity of a completely new variant made up of a combination of point mutations can be predicted with relatively good agreement to the experimentally observed activity. So this field obviously needs to be improved a lot more, but this is a new direction in which it is going. I will now discuss some questions of the based on these topics that we have discussed. One question is that what does the Ramachandran plot show? So the answer to this is the frequency of phi and psi values of the residues. So if you remember the Ramachandran plot, I talked about the phi and psi dihedral angles at the beginning of this class, and it shows the frequency of these values. So the white regions are where the values are completely disallowed. This dark region is where the values are completely allowed, and this light region is where it is the border region. So it is allowed, but it is not as much preferred as the dark regions. So definitely it is showing the frequency of phi psi values in the residues. It is not super secondary structure because it's only the secondary structure. It has nothing to do with hydrogen bond, and it has nothing to do with domains. If you remember, domains are basically what make up the quaternary structure. So this is only about secondary structure, so it has nothing to do with domains. So the correct answer is D, that is the frequency of phi psi values of the residues. The next question is asking which factors contribute to stability of the protein structure. So here the answer is all of the above. We looked into this hydrophobicity between amino acids which have aliphatic side chains like leucine, valine, isoleucine. There are stabilizing hydrophobic interactions, electrostatic interactions between positively and negatively charged side chains like acidic and basic side chains, as well as disulfide bridges between two cysteine residues. So CH2SH, CH2SH, the hydrogens are lost to make this disulfide bridge. So all these factors are stabilizing the protein. So the answer is D, that is all of the above. The next question is asking, which of the following is not a component of PCR reaction? So the answer to this is DNase, because in PCR, flanking primer is important. Flanking primer is basically you have the template, the initial primer that is attached to the 3' end of the template is the flanking primer. To that primer, all the other nucleotides is attached. So this is definitely present. DNTP is the deoxyribonucleotides, which are adding one by one to the flanking primer to do the extension step. So this is also important. C is the template DNA, which is the initial DNA, which is denatured to give the two primers. So this is also important. What is not required is DNase. DNase basically breaks the DNA into different parts. So definitely we don't need that for PCR reaction. So which is not a component is option D, that is DNase. The fourth question is asking the rational design of new protein molecules to design novel activity, behavior or purpose and to advance basic understanding of protein function is called as what? So the answer to that is protein design. So obviously it's the design of new protein. So it cannot be protein instability. It has nothing to do with protein unfolding or protein cleavage. So the rational design of new protein is generally done to design a new mutated protein which has a completely different activity or improved activity to the native protein. So these are all part of protein design, and they can be which is option A, and there can be different methods of protein design like directed evolution, rational design, or methods which combine the different methods. So that is all about protein design. That is option A. The next question is asking, what is de novo design in protein engineering? So here the answer is de novo design is engineering a new sequence based on prior sequence. So if you remember, in the de novo approach, we start completely from scratch. We first build the backbone based on other known sequences which have the secondary structure that we want, and we take fragments of the different secondary structure to design the new sequence based on the known previous sequences, and that is option B. Design of protein with a few mutated residues is called directed mutagenesis. So that is basically we have a previously known structure of the protein and we are just mutating a few residues to get the new protein. That is called directed mutagenesis. In de novo protein, we design a sequence completely from scratch, and that is done based on the knowledge of previous sequence structure relationships. So that is option B. The next question is asking, which of the following is the protein structure prediction method? So that is the homology modeling. So homology modeling consists of identifying homologous protein structures. Then the query sequence is aligned with the template sequence of the homologous protein structures. And then a model for the query sequence is built. So that is what is, and then that model can be refined and so on. That is what homology modeling is, and it can help predict the protein structure based on previously known structure sequence relationships. So it is option B. So homology modeling basically predicts the structure of an unknown sequence based on known sequence and structures. So it is a structure prediction method, that is homology modeling, option B. The next question is asking, ITASR structure prediction is based on what? So here the answer is multiple threading approach. So ITASR, the first step included threading, which refers to a bioinformatic procedures for identifying template proteins from solved structure databases, which have a similar structure or similar structural motif as the query sequence. So in threading, we take the entire unknown sequence and we find out portions of the sequence which belong to known structures. Multiple threading, we put all these known structures together and get a predicted structure for the unknown sequence. That is what ITASR is based on. That is the multiple threading approach. That is option C. The next question is asking, what is silent mutation? So this is basically when there is a change in nucleotide sequence with no consequence for the amino acid in the protein. So we saw it before also. If TTC is mutated to TTT, both are coding for lysine. So it is a silent mutation because there is no effect on the amino acid sequence in the protein. The next question asks, if we change one nucleotide, that is A to C, in the gene sequence of an enzyme, what is this called? So the answer is substitution. Changing one nucleotide. So if we change, suppose T to A, CTC, it becomes CAC. T is changed to A. That is called substitution. Deletion is deleting a nucleotide. Insertion is inserting a nucleotide. And large modification would include multiple changes. So just changing one nucleotide into another is called substitution. That is option B. And the last question asks, epistasis is a phenomenon observed in what? So the answer is genetics. Epistasis is basically when one gene completely masks another gene. So if you have two mice having uh, uh, genes where the capital B stands for more pigment, that is black, small b stands for less pigment, that is brown. Capital C stands for pigment or small c stands for no pigment. So you can split up the different genes and put them together again. And you can see that 4 out of the 16 possibilities will have small c, small c. So when both are small c, that is the small c is the recessive gene. So when both have small c, that means these 4 mice will have no color, no pigment, that is they will be albino mice. So the two genes, the capital B gene, this small c gene is completely masking the effect of the capital B gene. Because when two small c genes come together, they will mask the effect of the black or brown gene and it will cause a mice without any pigment, that is an albino mice. So that is what is epistasis, where the small c is completely masking the effect of the capital B small b gene. So that is a part of genetics, which is epistasis, and it's option B. I have two more questions. What is the requirement of the directed mutagenesis? So the requirement is you need the cloned DNA of interest. So whatever mutated DNA you have, that needs to be cloned so that it can be inserted in the plasmid. And an expression system is needed where the plasmid can be inserted and the protein can be expressed, which is generally E. coli. So you need both A and B, that is option C. And one second. Yeah, I think this is, what is the EPRO method we studied? It is to redesign proteins to increase or give specificity to native or novel substrates and cofactors. So option 1 is out because it is decreasing activity. No one wants that. Another, this is also out because it is designing a protein with few mutated residues, that is basically directed mutagenesis. And designing a new sequence based on prior sequences out, that is the de novo design method. So EPRO, what it is used for? It is used to increase specificity or to design completely new enzymes. That is the option C. So yeah, this is just someone had asked this doubt, so I've included this slide. So that is all about week 11. I'll end the class now, and as always, slides will be uploaded and the video will also be uploaded. Thanks.